Hello guys, nice to meet you. We are discussing today the second part of the digestive system, its abdominal part. Previous time we discussed the supradiaphragmatic part, which include oral cavity, pharynx and the esophagus of the digestive tract or the alimentary canal. And today we are discussing the parts of the alimentary canal which are located below the diaphragm. They include the stomach, the small intestine, and the large intestine, the colon. As you remember, the embryonic sources of gastrointestinal tract differ between the parts of the alimentary canal. Today we discuss this part, the gastrointestinal tract from the stomach to the colon. Its epithelium is simple because it develops from endoderm. All the epithelia which develop from endoderm are simple. They have only one layer of the cells. And here we have the simple columnar epithelium. Other tissues of the gastrointestinal tract in these regions include connective tissues, muscle elements, which are here only smooth muscle cells, no skeletal muscle cells in the stomach and inside the small intestine. So these muscular elements develop from the mesenchyme. Blood vessels also develop from mesenchyme, and we will have here the neural plexuses in the submucosa and between the muscle layers in the muscularis. And these neural plexuses, which include the ganglia, collections of cell bodies of neurons, and neural bundles of nerves, they develop from neural crest. And all the organs which are located in some body cavities are covered with mesothelium, which develops from the visceral layer of splanchtome, or the splanchnic layer of the lateral plate mesoderm. The functions of the portions of the alimentary canal include ability to motion, motility, secretion of some substances, digestion, which means breakdown of the food particles, food molecules, and absorption. Today we discuss this part of this picture. So in stomach, the motility includes the storage, peristalsis of food mixing and propulsion. So the stomach can distend and store the food until it is proceeding here. So here starts the initial digestion. Mixing and propulsion of food into the next portion, into the duodenum. The food goes into duodenum in portions, in small portions, through the sphincter, which is located between the stomach and the duodenum. The secretion in stomach includes secretion of hydrochloric acid, enzymes, pepsin and lipase, secretion also of mucus and some hormones. The hydrochloric acid and enzymes act on the food here and launch its initial breakdown. The mucus, which is secreted here, protects the epithelium of the stomach from the action of the hydrochloric acid and enzymes. The hormones are released into blood, not into the lumen of the stomach. The digestion here in stomach includes digestion of proteins, fats and carbohydrates. Here the digestion of proteins includes the breaking down of the large protein molecules into smaller ones, not in amino acids, just into the smaller protein molecules. And about the fats, fats inside the stomach are also breaking down mechanically into smaller droplets, and this is called emulsification. The carbohydrates start to digest inside the oral cavity, where the amylase and maltase are secreted by the salivary glands, and these sugars proceed their digestion inside the stomach. Absorption in stomach includes absorption of lipid-soluble substances, alcohol, and some drugs. Next, in small intestine, the segmentation and peristalsis takes place, mixing and propulsion of food particles. The food here is called the chyme. The chyme is the partly digest food plus the enzymes. What is secreted here in the small intestine? Bicarbonates, enzymes, mucus, bile, and hormones. Bicarbonates are secreted by the cells inside the duodenum, and these bicarbonates ions act upon the acid which comes with the chyme from the stomach and neutralize this acid, and the pH level changes from the acidic into the alkaline inside the duodenum, the first part of the small intestine. The enzymes which are secreted here are secreted by the cells of the small intestine itself, and also enzymes come here from the pancreas through the duct which opens inside the duodenum, and the pancreas gives almost all the necessary enzymes. Next, bile comes into the intestine from the liver and gallbladder. Mucus is produced by the glands of the duodenum and by the goblet cells in the epithelium of the small intestine. The hormones are released into blood, not into the lumen of the small intestine. Digestion. Almost all the molecules of the food are digested and absorbed inside the small intestine. So all the proteins are broken down here. The proteins break down first into polypeptide molecules, then into oligopeptides, then into dipeptides, and finally into amino acids, which are able to pass through the membrane of the epithelial cells of the small intestine and then into the blood capillaries of the small intestine. The fats emulsificated from stomach now here are broken down into the smaller molecules and also are digested here, and carbohydrates also are digested and absorbed here. So absorption here includes the absorption of peptides, amino acids, glucose and fructose, lipids, water, ions, minerals, and vitamins. Almost all the molecules from our food are digested and absorbed here in small intestine. In large intestine, in colon, the segmentation, mixing and propulsion of food proceeds, and the chyme is gradually becoming more solid to form the feces. The secretion here includes the secretion of mucus by the goblet cells, which are abundant here, and secretion of hormones, which go into blood. Digestion here includes the so-called bacterial digestion, because in our colon, in our large intestine, we have the symbiotic bacteria, which help us to digest all the molecules which were not digested previously in the small intestine. So these molecules are digested here with the bacteria. And what is absorbed here is water, ions, minerals, and vitamins. The structure of the gastrointestinal tract of the stomach, small and large intestine includes all the 4 layers. The innermost is mucosa, which includes the epithelium, lamina propria mucosa, and lamina muscularis mucosa. Second layer is submucosa. Third layer is muscularis or muscularis externa. And outer layer here is the serosa, because it includes mesothelium and underlying loose connective tissue. And we will have some patterns of relief here in stomach, small and large intestine, which includes macro relief, which consists of folds. The folds are always made up with 2 layers, mucosa and submucosa. So both of them make the folds. And the micro relief patterns will include the small pits in stomach, which are invaginations of epithelium into the underlying lamina propria mucosa. In small intestine, we will have the villi and crypts. Villi are evaginations of epithelium and lamina propria mucosa, and crypts are invaginations of epithelium into the underlying loose connective tissue of lamina propria. And in the large intestine, we will have only crypts. So, what changes when we go from the stomach to the distal portion of the colon is the changing of the so-called structural and functional unit of mucosa. Structural and functional unit means a kind of block from which some structure is made of. So in stomach, here in picture B, we will have the pit plus gland, structural and functional unit of the mucosa. In small intestine, we will have villus plus crypt, and in large intestine, we will have the crypts as structural and functional units of the mucosa. Also changes the presence and location of glands. In stomach, we have glands which are located in the lamina propria mucosa, above the lamina muscularis mucosa. In small intestine, we will have the glands only in the initial portion in duodenum, and here the glands are located in the submucosa. In the next portions of the small intestine, in jejunum and ileum, we will have no glands. In the colon, we also have no glands. Also differs the amount of muscle tissue and amount of number of layers in the lamina in muscularis externa. In the stomach, we have 3 layers: outermost longitudinal, next below it circular, and the innermost oblique layer. In small and large intestine, we have only 2 layers here: outermost longitudinal and innermost circular. So this is how it looks like in the histological preparations. So in esophagus, we had the stratified epithelium, and in stomach till the large intestine, we have the simple columnar epithelium. In stomach, we have the pits and glands. In small intestine, villi and crypts. And in duodenum also glands in submucosa. And in large intestine only crypts and no glands. So we start with the stomach. The function of stomach are deposition of food masses and their evacuation into the duodenum. The stomach has the folds, which are easily seen in anatomical preparations because they are kind of macro relief, and these folds help the stomach to distend when it is filled with the food. And while the food is stored here, it starts to be digested here, and it is then propulsed into the duodenum portionally with small portions. So here in stomach starts the mechanical and chemical processing of food. Mechanical is due to the very thick layer of the muscularis externa, which helps to move the food here. And chemical processing includes the non-specific digestion with the acid, with the chloride acid, and specific digestion of proteins with the enzymes, pepsin, and the specific protein for digestion of the milk proteins for babies. Initial digestion of lipids includes their emulsification here, making the large droplets of lipids more smaller droplets of lipids. Here is absorbed water, ions, glucose, alcohol, and some drugs. Aspirin, for example, is absorbed here. And here some hormones and biologically active substances are secreted. Hormones are secreted into the blood, and also in stomach the Castle factor is secreted, which is necessary for absorption of the vitamin B12 next inside the small intestine. So this Castle factor is produced here in stomach, but it will be necessary for digestion of vitamin B12 in the small intestine next. The types of relief here include gastric folds, large folds, or gastric ruggi in Latin, which are made up with both mucosa and submucosa. Also, we have here gastric fields, which are not seen in the large anatomical preparations, but are seen here. They are the parts of the mucosa separated with the loose connective tissue here, and gastric pits, the small invaginations of the epithelium into underlying loose connective tissue. This is how these types of relief look like in histological preparations. So this large is the fold, which includes mucosa and submucosa. This will be one gastric field and another gastric field. And these smallest invaginations are gastric pits. Here in the larger magnification you can see the gastric pits. In this picture, gastric pits and the gastric glands are seen, which open into the bottom of the gastric pit. So the general structure of the wall of the stomach is shown here. So here you can see the structural and functional unit of the mucosa, gastric pit, and gastric glands. They are located in the mucosa. Mucosa includes epithelium, lamina propria mucosa with blood vessels, and lamina muscularis mucosa. In submucosa, we will find large blood vessels and the lymphatic vessels, and also the nerve plexus, Meissner's plexus, which is necessary for innervating the blood vessels and glands. Next, muscularis includes 3 layers: innermost oblique, middle one circular, and outermost longitudinal. Outer layer is serosa. It's loose connective tissue with some blood vessels and nerves, and it is covered with mesothelium. In the structure of stomach, we define the three different in histological structure parts. The cardia, it's a small portion of the stomach just where the esophagus comes into the stomach. So this is the cardia. Also, we have the fundus and corpus here. They look the same in histological preparation, so we are discuss them as one, fundus and corpus. Fundus is this part of the stomach, and corpus is all the rest here. And third part, we have the pylorus. Pylorus is the portion of the stomach which is the closest to the duodenum. All the three parts have the same structural and functional unit of the mucosa, gastric pit and gastric gland. How they differ between each other? You should be able to differentiate between the portions of the gastric wall. So here are the criteria which you should use, and this is how it looks like. So first, the mucosa thickness. In the cardiac portion of the stomach, we have the thinnermost mucosa. It is thin. In fundus and corpus, it is thick, and in pylorus, it is also very thick. Next, depth of the pits. In the cardiac portion, the depth of the pits is 1/4 of the mucosa. So here is the pit, and here is the mucosa. In the fundus and the corpus, the pits are the smallest if you compare them between the parts of the stomach. So here they are just 1/6 of the mucosa. And in the pylorus, the pits are the largest, the deepest. They are 1/2 of the mucosa. Next, glands. Glands are simple in all three parts, but in the cardia they are branched. So in the picture of the histological section, we will see them as the oval and rounded in shape cuts of these glands. In the fundus and corpus, the glands are simple, non-branched or just slightly branched. They are tubular, so we can trace the gland from the pit till the lamina muscularis mucosa. They are very long and non-branched. And in the pylorus, the glands are simple branched with the convoluted or twisted secretory portions. So they also, as in the cardia, look like the circles and ovals. And finally, the cells of glands. We will discuss the gland in the example of the fundus, because the fundus and corpus has all cell types in the glands. But in the cardia and pylorus, the mucous cells predominate. They produce mucus, which helps to protect the epithelium from action of the acid and the proteolytic enzymes. Also, there are some additional differences, such as thickness of the muscular layer, muscularis externa, which is the thickest in the pylorus part, because the pylorus has the sphincter between the stomach and the duodenum. So here you can see the structure of the glands, and inside these three parts, in the cardia, the glands are simple, branched, tubular. In fundus and corpus, they are simple, non-branched or slightly branched, tubular. And in pylorus, they are simple, branched, with convoluted secretory portions or twisted secretory portion. This is how these preparations look like. So in the first one, we see the cardiac portion of the stomach with the pits and glands. Pits are about 1/3, 1/4 of the mucosa here, and the glands look like circles. In the second picture, we see the fundus with the smallest pits and the longest long tubular glands. Third picture also shows us the fundus with small pits and long glands, but this is different staining. This is the Congo red and iron hematoxylin, not hematoxylin and eosin. And finally, in the last one picture, we see the pylorus with the longest, deepest pits, and the glands, which look like some circles and ovals, with the predominating mucous cells, which look not stained here. So our first preparation will be the gastric fundus, and this will be your picture in your album. This is how it looks like. So it has mucosa, submucosa, muscularis, and serosa. In the mucosa, we have the pit plus gland, and we discuss next the structure of the gland, using the fundus as an example, because here the glands are the most well developed. They have all the cell types. This is how this preparation looks like. This is specific staining, Congo red and iron hematoxylin. Here you can see the fold, because it is made up with the mucosa and submucosa. In submucosa you can see large blood vessels here, and then muscularis starts. In the mucosa you can see the pits and glands. Another example, here you can see another pattern of the relief. This is not a fold, this is the gastric field, because it is made up only with the mucosa. And the submucosa doesn't make this type of relief. These are gastric pits, these are gastric glands. Lamina muscularis mucosa is the border between the mucosa and submucosa. In submucosa you can see some large blood vessels, bundles of nerve fibers, and this structure is the lymphatic nodule or lymphoid follicle, which performs the immunity function here. Another example and some other example of this preparation. And now we discuss the fundic gland of the stomach. So in this picture you can see the gland, and it opens into the bottom of the gastric pit. So here is the bottom of the gastric pit. The gastric pit is covered with the columnar epithelium, which is mucus producing. So this is the surface epithelium made up with the surface epithelial cells producing mucus. The gland consists of the isthmus, or the opening of the gland. It is the transitional zone between the epithelium of the gland and epithelium of the gastric pit. Next, the neck zone, neck of the gland. Next, the body of the gland. Most of the gland is its body. And the bottom, or the base of the gland. And here you can see the cells. So these are the cells of the gastric pit, the surface mucus producing epithelium. In neck, we have the stem cells, which are the stem cells both for the epithelium covering the pits and epithelium of the gland. So here the cells are proliferating, dividing, and next they differentiate either into surface epithelium, and in this case they move upwards, or into the cells of the gland, and in this case they move downwards and differentiate into the cells of the gland. Also in this region in the neck we have the mucous neck cells, which produce mucus. Next, in the body of the gland we have the parietal cells. They are very large, and their basal part kind of go out from the line of the gland, but it stands upon the same basement membrane as all other cells here. These parietal cells produce protons, H plus ions, and chloride ions, which together make the chloride acid of the stomach juice, gastric juice. Also, these cells produce the Castle factor, which is essential for absorption of B12 vitamin inside the small intestine. All these substances are released through the apical surface of the gland into the lumen of the gland, from the apical surface of the parietal cell into the lumen of the gland. And also this parietal cell produces the bicarbonate ions through their basal part, through the basement membrane. Next, chief cells, here and here also, they produce the pepsinogen. This is a not active enzyme which goes into the lumen of the gland here, where it meets with the chloride acid and converts into the active form, into the pepsin. So the pepsin is the proteolytic enzyme, which means that it breaks down the proteins into the smaller molecules, polypeptides. Next, mucous cells here. They are scattered between the chief and the parietal cells. They produce mucus. And in the bottom of the gland, we have almost only endocrine cells. Endocrine cells produce the hormones which can go into the blood capillaries here, or they can act upon the nearby cells. For example, they can act upon the parietal and chief cells, making them to produce their substances more or less actively. So endocrine cells regulate the activity of other cells in the gland. This is how these cells look like. In the electronograms, these are not really electronograms, these are just pictures. So we have 5 types of cells here, plus Campbell cell, which means the stem cell. It is the stem cell for all 5 types of the cells here. You can distinguish the Campbell stem cell because it has very few cytoplasm and very few organelles in it, because it is not differentiated yet. Next, mucous cell produces mucus, and you can see the granules filled with non-stained mucus. It is whitish looking non-stained in its apical portion, because it releases mucus through its apical portion into the lumen of the gland. The mucus is the glycoprotein molecule. It means that it consists of the protein and the sugar parts. You should be able to describe the organelles which are needed to produce the substances which are released by the cells. So if the cell produces mucus, you should say that mucus is the glycoprotein, so the cell needs to have the rough endoplasmic reticulum to produce the protein part of the molecule and the Golgi complex to produce the carbohydrate part of the molecule. So the mucus producing cells have rough endoplasmic reticulum and Golgi complex to produce mucus. Next, surface epithelial cell. It covers the pits of the stomach, so it lines the stomach from the inside. It produces mucus also, so it has rough endoplasmic reticulum and Golgi complex. And it has the granules of the mucus in its apical surface. It constantly releases the mucus through its apical surface to have the mucus at the surface of the cell, and this mucus will protect the cell from acting of the acids and proteolytic enzymes. Next, chief cell produces the enzymes in inactive form, which are called zymogenes. Zymogenes are inactive enzymes. The cell cannot produce the enzymes in active form because in this case they would digest the cell itself. So the cell produces non-active enzymes which go into the lumen of the gland. There they meet the acid and become the active enzymes. So chief cell produces the pepsinogen and the inactive form of lipase. Enzymes are the proteins themselves, so if the cell produces proteins, it is exocrine cell, so it has the granules with the proteins in its apical part, and also it has very well developed rough endoplasmic reticulum and Golgi complex. Next, parietal cell. It releases ions H plus and chloride minus protons and chloride ions, which together make the chloride acid. And the cell has very strange looking invaginations of the apical membrane into the cell, which increases the surface of the cell to have the ability to place more ion channels and ion pumps inside this membrane to produce the ions from the apical surface. And because the cell has a lot of ion pumps, which are ATPases, they use ATP to make their job, so the cell also has a lot of mitochondria in its cytoplasm. And endocrine cell here, it releases the hormones through its basal part, through its basement membrane, into the loose connective tissue and into the blood capillaries. So it has the secretory granules in its basal part, below the nucleus. Again, the main cells of the gastric gland, the chief exocrine cell and the parietal exocrine cell. Chief endocrine cell produces the zymogen, which is the inactive form of the enzyme. So it has the granules of zymogen in its apical surface. And it has well-developed rough endoplasmic reticulum and Golgi complex. And here the parietal exocrine cell, it has the invaginations of the apical membrane, which makes the intracellular canaliculus. Canaliculus is the Latin word meaning the small channels. So these small channels are invaginations of the membrane, and also you can see a lot of microvilli in the wall of these canaliculus. They also help to increase the surface area of the apical membrane of this cell. Why it is necessary? It is necessary to place more ion channels and ion pumps inside this membrane of the cell. So how it works? It takes the water molecule and the CO2 molecule. Next, it uses the enzyme carbonic anhydrase to make the acid HCO, H2CO4, and then it splits into the bicarbonate ion and H plus ion proton. Bicarbonate ion goes from the cell through its apical surface to the underlying loose connective tissue through the anion antiporter channel, which means that bicarbonate ion goes out from the cell, and at the same time the chloride ion goes inside the cell. And next, the ATPases use the ATP molecule to pump the H plus ions from the apical surface into the lumen of the gland. And also the chloride ions go through the channels also inside the lumen of the gland. And here the chloride and the proton meet together to form the chloride acid. And here you can see the information about the endocrine cells of the gastric gland. Endocrine cells are located mostly in the bottom of the gland here. And here is the list of some of the cells. So you should be able to explain the function, for example, of the G cells, which release the gastrin, which acts upon the parietal and chief cells and stimulates them to release their substances, to release the hydrochloric acid and to release the pepsinogen. And the D cells release the somatostatin, which also acts upon the parietal and chief cells and make them to which inhibits their secretion. So G cells stimulate parietal and chief cells, and D cells inhibit them. They make the regulation of the function of the glands. So in this picture you can see how these cells are distributed inside the epithelium of the stomach. So these are the pits. They are covered with the surface epithelial cells, which produce mucus. They have the secretory granules in their apical part, and rough endoplasmic reticulum and Golgi complex. Next, in the neck of the gland, where the gland opens into the bottom of the gastric pit, we will have the Campbell cells and also mucous neck cells. And next, in the body of the gland, we will have the mucous, parietal cells and chief cells, and in the bottom of the glands we will have the endocrine cells. This is how it looks like in the histological preparation. This is the uppermost part of the mucosa, and this is the middle portion of the mucosa, you can see here the glands of the fundus of the stomach. In the first picture, this is the gastric pit. It is covered with the surface epithelium, with the surface epithelial cells. They have the pale, transparent looking cytoplasm because it is filled with the granules of the mucus. Next, this is the neck of the gland, where the gland opens into the bottom of the gastric pit. Here you can see a lot of cell nuclei. These are the cell nuclei of the cambial cells. They have very few cytoplasm, so this cytoplasm is not seen. You can see here only the cell nuclei, so you can say that these are stem cells. Next, we will have the cells of the body of the gland, mucus, parietal, and chief cells. Mucus cells are those which have the pale, not stained cytoplasm around the nucleus. Nuclei in this staining are violet. So these are the glands, a lot of glands here. And for example, this cell can be a mucus cell because it has a nucleus, a violet dot here, and it is surrounded with non-stained cytoplasm. Parietal cells are the most well seen here. These are the largest ones and they are red in color. They are red because of mitochondria. Chief cells have the bluish cytoplasm because of rough endoplasmic reticulum. So you should find the cell with the nucleus and bluish looking cytoplasm. These are chief cells. And endocrine cells are not seen in our histological preparations. They are present, but we cannot define them. Here you can see the uppermost part of the gastric mucosa with the gastric pits here and the gastric glands, their initial parts with their necks and the part of their bodies. And here is the lumen of the stomach. It has a very low pH level because it is filled with gastric juice with chloride acid, which is produced by the parietal cells with red cytoplasm. Also, gastric juice includes pepsin, enzyme produced by chief cells, and lipase, also from chief cells. But here you can see the layer of the mucus, which covers the epithelium. It is produced by the surface epithelial cells themselves, and the pH level here is the same as inside the cells. It is normal pH level, about 7. And it is normal because the cells release, the surface epithelial cells release the bicarbonate ions inside this mucus, which neutralize the acid in this layer of the mucus to protect the cells from the action of the acid. So here you can see the glands inside the lamina propria mucosa. These large red are parietal cells, and between other there are chief cells with blue cytoplasm, mucous cells with pale cytoplasm, and there should be also endocrine cells not seen in this picture. The cells with flattened elongated nuclei are the endothelial cells of the capillaries of the lamina propria mucosa. Here you can see some blood vessel here. So the glands are tubular, simple, and they are very tightly packed here. And it is hard for us to find the lumen of the glands. how we can find the lumen? We should find the layers of loose connective tissue with blood vessels. Here one layer and here another one layer. So this will be one gland between the loose connective tissue. So the lumen should be somewhere here in the center of this gland. Another example here, for example, cells of loose connective tissue, and here also, and between them the gland. So the lumen should be here. It is closed because it becomes visible, it becomes open only when the cells are producing their substances, when they are actively producing the enzymes and the chloride acid and the mucus. And this happens only when we eat something. When we don't eat anything, then the cells do not produce their substances, and we cannot see the lumen of the gland. Another examples of this preparation, and this is also gastric fundus, but stained with hematoxylin and eosin. You can see here all the wall, the structure of all the wall here, with the mucosa, with gastric pits and gastric glands. This is lamina muscularis mucosa. Below it, submucosa with large blood vessels here. Muscularis externa with the innermost very thin layer, very thick middle layer, and outermost longitudinal layer. And the outer layer of the wall is the serosa. This is cardial division of the stomach, because the pits are deeper than in fundus, and the glands are not so elongated, they are branched, so they look like circles here. And as you can see here, the cells, the most of the cells are mucous cells with pale transparent cytoplasm. Lamina muscularis mucosa, submucosa with large blood vessels, and muscularis externa. The structure of gastric pylorus is shown here in this picture. The main difference of the gastric pylorus between this portion, between pylorus and other portions, is the depth of the pits. The pylorus has the deepest pits. This is how it looks like in the preparation. So these are very deep pits of the mucosa. These are the glands, lamina muscularis mucosa, submucosa, and muscularis externa, covered with the serosa. You can see here the second difference of this region of the stomach, very thick layer of the muscularis externa, with the most well-developed middle layer, circular layer, because it makes here the sphincter between the pylorus and the duodenum. At high magnification, you can see here the gastric pits, very deep gastric glands, which look like circles, and they consist mostly of the mucous cells. The stem cells are located between the bottom of the pit and the neck of the gland. So here should be the stem cells, both for epithelium of the gastric pit and epithelium of the gastric glands. Another examples. And this is next preparation, transition duodenum. Here you should be able to define between the stomach and the small intestine. Its first portion, duodenum. Duodenum here is in the left part of the picture. Here is duodenum. In the right part we have the stomach. What changes when we go from the stomach into the duodenum? The major difference is the structural and functional unit of the mucosa. In stomach, gastric pits and gastric glands change into the villi and crypts of the duodenum. Villi and crypts. Second difference, the location of glands. Both structures have the glands, the stomach and the duodenum, but their location differs. In pyloric part of the stomach, we have the glands in the lamina propria mucosa, and they are located above the lamina muscularis mucosa. In duodenum, we have the glands in submucosa. They are located below the lamina muscularis mucosa. You can trace the lamina muscularis mucosa and see how the location of glands differs. And the third difference between these structures is the thickness of the muscle layer of the muscularis externa. It is very thick in the stomach. We can see here only its part, and it becomes thinner in the small intestine. So in this region we have the sphincter between the stomach and the small intestine. So here you can see the differences between the pylorus, structure and functional unit of mucosa, location of glands, also cell types of the epithelium lining, which are best seen in high magnification. In the epithelium of the gastric pits, we have only one type of cells, the surface epithelium mucus-producing cells. But in the epithelium covering the villi, we have two major types of cells. The cells with microvilli with brush border and goblet cells. Goblet cells look like small whitish dots inside the epithelium. No whitish dots you see in the epithelium of the stomach, because all the cells here are the same. Thickness of the tunica muscularis and number of layers in the tunica muscularis also. In stomach we have 3 layers, outermost, middle, the thickest, circular, and innermost, oblique, very thin. In small intestine, in duodenum, we have 2 layers, outer longitudinal, inner circular. In this picture, the stomach is in the left picture, duodenum is in the right one. So here you can see in the stomach pits and glands. In duodenum, villi and crypts. The glands are located in stomach above the lamina muscularis mucosa, so they are located in lamina propria mucosa, and in intestine the glands are located below the lamina muscularis mucosa, so you can say that they are in submucosa. You can see in the epithelium covering the villi in the duodenum goblet cells. Other cells are pinkish in color, and in the epithelium covering the gastric pits you can see that all the cells are the same, because they are all one cell type, surface epithelial cells. Another example of this preparation, transition between stomach and duodenum. Here stomach is in the left part and duodenum is in the right part, because the major difference is the structure and functional unit of the mucosa and location of glands. Here in stomach the glands are located above the lamina muscularis mucosa, and in the duodenum glands are located below the lamina muscularis mucosa. Next, the general structure of the small intestine includes all the 4 layers, mucosa, submucosa, muscularis, and serosa, and the structural and functional unit of the mucosa is villus and crypt. One villus, many villi. Types of relief here include largest one is fold. Fold is made up with the mucosa and submucosa. You can see that this is the submucosa because you can see here large blood vessels. Villi and crypts. Intestinal villi are not so numerous as the crypts, so the crypts are much more numerous as the villi. So in every villus we have the epithelium and lamina propria mucosa with blood vessels and lymphatic capillaries here. They are blind-ended, so this is the beginning of the blood of the lymph capillary. You can see here the epithelium of the villus and of the crypt. Here is the villus in the right part. In the left part you can see the crypt, and in this epithelium, as in the epithelium of the stomach, we have also 5 types of the cells. The most numerous cell here is the cell with the brush border. The second one is the goblet cell, mucus-producing cell. They both are found in the epithelium of the villus and in the crypt also, but in the crypt we have additional cell types: the endocrine cells, which produce and release hormones, and the Paneth cells, which are located in the bottom of the crypt. Also in the crypt we have the stem cells, which have the mitotic figures in them. You can see here one intestinal villus with the epithelium and lamina propria mucosa, and in the center of the villus you can see large lymphatic capillary. Also smooth muscle cells are present in the villus. They contract when the food molecules are absorbed, and it helps the food molecules to pass downwards from the villus into the underlying submucosa. Sorry. In the picture you can see the cells of the crypt. In the bottom we have the Paneth cells, or exocrine cells with acidophilic granules. Next we see the cells with the brush border, absorptive enterocytes, goblet cells, and stem cells. The functions of the cells: columnar absorptive enterocytes produce the enzymes which are essential for the last stage of the digestion of food, for breaking down the food molecules from dimers into monomers. For example, dipeptides into amino acids. And after these amino acids can be absorbed by the cell. The second function is the absorption itself, which means the passing of the molecule from the lumen into the cell and then through the cell, and release this molecule through the basal portion into the blood capillary of the loose connective tissue. Also, the cells with brush border can can uptake the antibodies, immunoglobulins produced by the plasma cells in the lamina propria mucosa, and release these antibodies into the lumen of the intestine. Goblet cells produce mucus. Mucus helps the digestion because it covers the surface of the epithelium and it is sticky, and the food particles and enzymes are trapped inside this sticky layer of mucus, and they come in touch together here, and the processes of digestion can proceed. Paneth cells, which are located in the bottom of the crypt, produce the defensive molecules for defense against bacteria, such as defensin, lysozyme, and some other defensive molecules. Stem cells differentiate in all the 5 cell types here. This is how the Paneth cells look like. They are located in the bottom of the crypt, and they have acidophilic granules inside their apical portion. In duodenum, unlike other portions of the small intestine, in ileum and jejunum, here in duodenum we have the glands, duodenal glands, or Brunner glands, which are located in submucosa. This is the electronogram of the main cell type of the small intestine, absorptive enterocyte with the brush border, which consists of microvilli. Brush border is the layer made up with the very well developed microvilli on the apical surface of all the absorptive enterocytes. You can see here the cross section of microvilli. They have the actin microfilaments inside them, and they are covered with glycocalyx, grayish looking. Between the cells you can see the complex of junctions, including tight junctions closest to the apical surface, and then belt desmosomes, some other types of contacts, and the cells lie upon the basement membrane, and below there is the loose connective tissue with blood capillaries. Here you can see at the very high magnification the apical surface of some cells with the brush border. You can see here the parts of the 3 cells, because you can see the border between 2 of them here, and the third one is here. These are the microvilli. They are covered with the glycocalyx. Glycocalyx serves for 2 main functions: for reception and for digestion. The membranous stage of digestion takes part here, because the enterocyte produces the enzymes for this last stage, and these enzymes are trapped inside this glycocalyx layer, and here they break down the dimers into monomers to make it possible to absorb these monomers by the enterocyte. Types of digestion or stages of digestion in small intestine include luminal, parietal, membranous, and intracellular. Luminal means that it takes part in the center of the lumen. Parietal means that it takes place closer to the mucosa. Membranous means that it takes place near the membrane of the epithelial cells, and intracellular means that it is inside the cells. What does it mean? Luminal. Here the large polymer molecules are broken down into the smaller molecules. with the help of the pancreatic enzymes which come into the duodenum from the pancreas, and these enzymes with the help of the duodenal glands are forming the so-called floccules, where the enzymes meet their substrates, the food particles which they digest. Next, parietal is closer to the cells, and it takes place inside the lumen of the mucus covering the epithelium. Here, the hydrolysis of oligomers takes place. The oligomers are broken down here into dimers, and this is with the help of the enzymes of intestinal juice, which include the enzymes coming from pancreas and the enzymes produced by the enterocytes themselves. Next, membranous stage of digestion is inside the glycocalyx layer. Here, dimers are hydrolyzed into monomers, and monomers are absorbed by the enterocyte. And this is with the help of the enzymes produced by this enterocyte itself. And finally, intracellular stage is breaking down of the absorbed molecules with the help of the lysosomes inside the enterocyte. So here you can see some lysosomes. Enteroendocrine cells in the epithelial layer include two subtypes. One of them enteroendocrine cell of the open type, and second of the closed type. In the first case, you can see that the apical surface of this cell reaches the surface of the epithelium. It reaches the lumen of the intestine. In the second type, in the type of the endocrine cell of the closed type, it doesn't reach the surface. But you can say easily that these two are endocrine cells because they have secretory granules in their basal part, below the nucleus. It means that they are going to release these secretory granules through their basal surface into the blood capillaries. In duodenum, we have the duodenal or Brunner glands, which are compound tubular alveolar branched, and they are located in submucosa, below the lamina muscularis mucosa. In high magnification, crypts of the mucosa, lamina muscularis mucosa, and secretory portions of the duodenal glands in submucosa. The cells of these glands include mucous cells, endocrine cells, and the stem cells. The functions of this gland includes production of mucus with bicarbonate ions. These bicarbonate ions act upon the acid which comes from the stomach and neutralize this acid and makes the pH level first neutral and then alkaline. So the pH level here switches from the acidic to alkaline. Next, the mucus produced by the duodenal glands increases the viscosity of the layer of mucus, making it easier for enzymes to stick into this mucus and make their job. Also, duodenal glands help the flocculi formation and increase absorption of duodenal juice to the layer of mucus. So you can see here that duodenal glands do not produce enzymes. They just produce mucus, rich in bicarbonate ions. All the enzymes come from pancreas or are produced by the cells with brush border. This is how the digestion process in small intestine happens. So from the stomach, where the pH level is about 1, 2, it's very acidic, the chyme particles, the particles of partly digest food, come into the duodenum, where they are surrounded with the mucus produced by duodenal glands, and this is called the flocculi. So flocculi include the chyme particle surrounded with the mucus produced by duodenal gland, and here also in this mucus the enzymes from the pancreas stick. They are trapped inside this mucus. So here in this floccula, the chyme, the food molecules and enzymes for their breaking down meet together with the help of mucus, and this increases the rate of the digestion. In stages, here the same. First, the chyme comes from the stomach. Second, the chyme is covered with the mucus produced by duodenal glands. 3. The enzymes also come into this flocculi. Next, number 4, this chyme, these food molecules are broken down into smaller particles with the help of these pancreatic enzymes, and then these smaller molecules go into the parietal layer of mucus, which covers the epithelium, and here the parietal digestion takes part. Also with the help of the pancreatic enzymes, where the polymers are broken down into oligomers. Then the smaller molecules go closer to the epithelium, into the glycocalyx of the cells with brush border, where the membranous digestion takes place, and finally they go inside the cells. The smallest molecules, monomers such as glucose, amino acids, come into the cytoplasm of the cells with the brush border. Absorption is the transportation of end products of the hydrolysis from the lumen of the intestine into the cells, and next into the loose connective tissue and into the blood or lymphatic vessels. All the digested food molecules except fats come into the blood capillaries. The digested fats come into the lymphatic capillaries. This is because all the other molecules except of fats are very small. They can easily pass through the wall of the capillaries, which here are of the second type, the visceral type, fenestrated ones. So these molecules come into the blood capillaries. But the digested fats are very large molecules. They are digested in the form of so-called chylomicrons. They are so large that they cannot pass into the blood capillaries, but they can pass into the lymphatic capillaries. So the fats are digested with the lymphatic system. This is the example of the preparation of the duodenum. You can see here the villi and crypts and duodenal glands in the submucosa. Another preparation, here for example you can see, you can define how the gut was sected. What is the plane of sectioning? This can be asked from you, so you should be able to define the plane of sectioning. You should look into the outermost layer of the muscularis externa. This is the muscularis externa. It has two layers, outermost layer and inner layer. Outer layer is longitudinally longitudinal. Inner layer is the circular. If you see that the cells in the outer longitudinal layer are longitudinally sected, it means that you can see their elongated cell nuclei of the smooth muscle cells, then you can say that all this layer was longitudinally sected, and all the gut, all the intestine was sected longitudinally. In other examples, in the villus, you can see the smooth muscle cells here and the lymphatic capillary in the center. In the epithelium, you can see here the cells with the brush border and the goblet cells. Goblet cells have the non-stained apical portion because it is filled with the granules of the mucus. Mucus is not stained with the hematoxylin or eosin. The nucleus of the goblet cell is in its basal portion, and the cells with the brush border have the pinkish cytoplasm because of a lot of cytoplasm proteins and mitochondria. Another examples here. For example, somewhere you can see also the intraepithelial lymphocytes that go into the epithelium, for example here, to perform the immunologic surveillance. Jejunum, unlike the duodenum, lacks the glands. So if you look into the submucosa, you can see here no glands. So this is the major difference between the duodenum and jejunum, the absence of glands in the jejunum submucosa. Other differences include the shape of villi. In duodenum, the villi are shorter and thicker than in the jejunum. In jejunum, they are longer and thinner. And also number of goblet cells. The number of goblet cells increases gradually from the stomach towards the colon. This is the preparation of jejunum. It is the cross section, because if we look at the muscularis externa, we can see that the outer layer was cross-sected and the inner layer is longitudinally sected. So if the outer layer is cross-sected, then all the gut was also cross-sected. Here is the mucosa, submucosa, muscularis, and serosa. In mucosa you can see a lot of villi, here the crypts, and lamina muscularis mucosa. In submucosa, which is reddish here because of the collagen fibers, you can see also some large blood vessels. Another example, here in the serosa you have a lot of adipose tissue. Amount of adipose tissue increases in fat persons and also in aged persons. Another example, villi and crypts in mucosa. No glands in submucosa. The section was transverse cross-section because of the cross-sected smooth muscle cells in the outer layer of muscularis. Here at the high magnification you can see the mesothelium covering the intestine from the outside. So if you see the mesothelium, you can say that the externa is serosa here. The regeneration of epithelial tissues in stomach and in small intestine goes from the stem cells, which are located in the neck of the glands in stomach and in the lower part of the crypts in small intestine. After proliferation, the cells start to differentiate and to move upwards or downwards. And if they move upwards, they differentiate into the cells of the villi in small intestine or into the cells of the pits in stomach. These cells are in contact with the content of stomach and small intestine, which includes acid and some enzymes, and the cells of the lining epithelium have smaller lifespan than the cells of the crypts in the small intestine and of the glands in the stomach. So, for example, the endocrine cells at the bottom of the gastric glands can live up to 2 months, but the cells of the gastric pits live up to 7 days, usually about 3-4 days, and then they die and are replaced by newly produced cells from the stem cells in the neck of the gland. Next, colon, the large intestine, has the structural and functional unit of mucosa, crypt. So in the mucosa you can see here only crypts. They are invaginations of the epithelium into the underlying loose connective tissue of the lamina propria mucosa. The types of relief here are folds, made up with the two layers, two tunics, mucosa and submucosa, and crypts made up only with the mucosa, epithelium and lamina propria mucosa. Here you can see the structure of the mucosa of the colon. Epithelium includes the stem cells, which are located in the lower part of the crypt, the goblet cells with non-stained apical portion. They are the major cell type here, and between them, with pinkish looking cytoplasm, we have the enterocytes with brush border for absorption. Also, in the lower part of the crypt, we will have the endocrine cells. The function of the colon is to absorb the rest of the water and ions and to absorb some molecules produced by bacteria which colonize the colon, symbiotic bacteria. Also in the colon, the food masses, the chyme, is gradually lacking water because water is absorbed here, and the chyme becomes more and more solid to form the feces. This is why the colon has so much goblet cells. They produce mucus that covers the epithelium and protects the epithelium from the mechanical disruption by the solid feces masses. So here you can see the cells of the crypts of the colon. They include the major cell type, the goblet cell, also absorptive columnar epithelium cell between the goblet cells, the stem cells, and the endocrine cells. Also, we have in the large and in the small intestine both the so-called Peyer's patches. They are the patches where lymphoid tissue makes not only the single follicles, but is located where the lymphoid tissue is very, very abundant. And in such areas, no crypts and no villi in the small intestine are present. So in the Peyer's patches, you will see a lot of lymphoid tissue with a lot of lymphoid follicles in the lamina propria mucosa covered with the epithelium which doesn't make any types of relief, just flat epithelium. And in such epithelium, both in small and large intestine, you have additional cell type of the epithelial cell, so-called M cell. The function of such cell is to provide antigens from the lumen of the small and large intestine to the underlying cells of the lymphoid follicles, to the antigen-presenting cells, which then present this antigen to T helpers, which launch the immune reaction, which ends in production of antibodies by plasma cells, and these antibodies are taken from the loose connective tissue with the absorptive columnar epitheliocytes and are released through their cytoplasm into the lumen of the intestine. And this happens both in large and small intestine. This is the preparation of the colon. This is the longitudinal section, because if you look at the muscularis, you can see here that the outer layer is longitudinal sected. So this is the longitudinal section. In the mucosa, you can see only crypts. You can easily define the crypts from the pits and glands of the stomach and from the villi and crypts in the small intestine, because you can trace every or almost every crypt from the surface till the lamina muscularis mucosa. So you can say that here you can see only crypts. The second difference from the epithelium of the small intestine is the abundance of the goblet cells, which are non-stained here, which are looking with the whitish bubbles in the epithelium. Also, here you can see the outer layer of the muscularis externa, but as you should know from the anatomy, in the colon we have only inner layer of muscles continuous, but the outer layer, longitudinal layer, is incontinuous. It makes three ribbons which go along the colon. So the outer layer is incontinuous. Another example here, mucosa with crypts, muscularis mucosa. Below it here you can see a large lymphoid follicle. You can find in the colon a lot of lymphoid tissue because colon is colonized by the bacteria. So they are called symbiotic, but they can become pathogenic if the immune system will not defend the organ from the bacteria. So lymphoid tissue protects the colon from the bacteria from the lumen. And you can see here both inner and outer muscular layers in the muscularis. Another example here, another here. You can see here that the lymphoid follicle is so large that it took both the submucosa and mucosa also, and the lamina muscularis mucosa here is incontinuous. Another example. Here we can see the example of the intramural ganglion in the muscle layer, in the muscularis externa. Large blood vessels in submucosa. Also some nerve plexuses can be found in submucosa, but not seen in this particular picture. At higher magnification, you can see here the bottoms of the crypts. Most of the cells here are goblet cells because their cytoplasm is not stained. It is filled with the granules of the mucus, not stained with hematoxylin and eosin. Some intraepithelial lymphocytes with small rounded nuclei, lamina muscularis mucosa, and underlying loose connective tissue. You can say that this is a large and not small intestine because of the prevalence of goblet cells in the crypts. In small intestine, in crypts, we will have the Paneth cells, the cells with brush border, and endocrine cells. And our final preparation is the appendix. Appendix is the part of the large intestine. It is blind-ended. It has also 4 layers: mucosa, submucosa, muscularis, and serosa. But its major difference is the very well-developed lymphoid tissue inside it. And it is so well developed that it fills both the mucosa and submucosa, and you can see here no border between the mucosa and submucosa, because lymphoid tissue fills all the space here. No place for lamina muscularis mucosa. The major differences between the appendix and the colon are, first, as I said, a lot of lymphoid tissue with the follicles. Lymphatic follicles, as you know, have the germinal center and the crown. In germinal center, the antigen-dependent differentiation of B lymphocytes takes place, and as a result, they differentiate into the plasma cells, which located here just below the crypts, and they produce the antibodies, which are released into the lumen of the appendix, and here they fight against the symbiotic bacteria. And the second type of cells, which are produced as a result of the antigen-dependent differentiation of B lymphocytes, are the B memory cells, which stay inside the crown. The T zones are between the lymphoid follicles here for T lymphocytes. So a lot of lymphoid tissue first, the absence of lamina muscularis mucosa here, so it's not easy for us to say where the mucosa ends and where the submucosa starts, but you can find the bottoms of the crypts, and they will mark for you the border between the mucosa and the submucosa. So here should be the border between the mucosa and submucosa, where the bottoms of the crypts end. Few crypts, they are fewer here in appendix than they are in colon. They are much shorter, so they are not so well developed. Also, here not so many goblet cells are present in this epithelium, and the last difference is the structure of the outer layer in the tunica muscularis. Here it is continuous. In the appendix, the outer layer is continuous, unlike the colon, where it forms the 3 ribbons. Another examples of this preparation: a lot of lymphoid tissue, poor developed crypts, and not seeing the border between the mucosa and submucosa, and continuous outer layer in the muscularis. Another example of preparation: crypts are seen here, some adipose tissue in submucosa, 2 layers of the muscularis. Crypts, lymphoid follicles, submucosa, muscularis, serosa. Another examples, and that's all. So the major task for you in this topic is to be able to differentiate between the portions of the gastrointestinal tract, and the major difference is their structural and functional units. So this is the main information that you should remember: the structural and functional units of the large, small intestine, and in the stomach, and all the other differences also. So that's all. Thank you for your attention. Have a good night.